about our work to try to develop therapies to both treat and prevent prion disease. And as you already heard, this is a personal mission for my husband and I. So it's now been about 12 years since we changed careers, um, following the experience of losing my mom to prion disease. And it merits saying again, just how rapid this disease is and how completely shocking. My mom went from being a healthy 51-year-old to being on life support in the matter of just 4 months. So it was a completely shocking and unexpected experience for our family, and even more so when we learned that her disease had been genetic and that I was at 50-50 risk of having inherited her mutation. So this is a photo of my husband Eric and I. We made the decision almost right away to get predictive genetic testing. I tested positive. Prior to this, I had trained as a lawyer and Eric had trained as a transportation engineer. So we were not scientifically trained at all. But learning that I did have my mom's mutation, what ended up being extremely motivating for us and catalyzed a big change in our lives. So today we run a lab at the Broad Institute, as you heard, in Cambridge, Massachusetts. And really, it is a mission-oriented lab. Our goal is to get meaningful drugs to patients in our lifetimes. That turns out to involve, number one, a lot of patients. Number two, a lot of other activities in order to make meaningful trials possible. So part of what I hope to share today is what this journey has looked like and what preclinical drug development actually looks like from the inside. I know that we are hearing from patients and families all the time, and one of the hardest things to convey is that uh, we are making, I am optimistic about all the progress that we're making. And nevertheless, it does take so long and there are so many steps. And why? Why does it have to be that way? So here's a photo of our lab. We're 14 people. Um, and I'm going to take us back in time 10 years to the first conversation that we really ever had with a drug company that would end up being our first partners in developing a drug for prion disease. So pictured here is Jeff Carroll. He's one of our most important mentors. Jeff is at risk for Huntington's disease. So he carries a mutation that he inherited from his mom, and he's also a Huntington's disease researcher. So you can imagine that he's a hero to us. And he was the person who first said, 10 years ago, that Eric and I needed to meet the people at Ionis Pharmaceuticals in Carlsbad, California. We actually ended up visiting them together and spent a whole afternoon with their neuroscience team, pitching them on prion disease. Ionis, then and now, is focused on a really specific technology some of you might be familiar, which is antisense oligonucleotides, or ASOs. And this drug type is capable of binding a specific RNA. So it's sort of sequence specific to that RNA molecule that encodes a certain protein, and it can trigger that RNA to be destroyed. So I'll talk more in a little bit about why this is the right therapeutic strategy for prion disease. At any rate, this first conversation led to what's been a 10 years and counting collaboration with Ionis. And I'm going to show of many, many, many experiments that were done over the years, one survival curve. So what you're looking at here, for those who aren't familiar with survival curves, is a group of mice, actually a few different groups, all of whom were injected with prions on day 0. And this is how we model prion disease in animals. The animals then go on to develop fatal prion disease. So this is a disease of mammals, not just people. And we can actually watch the whole disease process happen in mice. They incubate prions, so prions are kind of replicating in their brains. Then they develop symptoms, and then they actually die of full-blown terminal prion disease. So you can see the gray line is animals that did not receive any therapy. And it's really right around 150 days, maybe 5 months after prion inoculation, that these animals reach the end of their illness. Like they die of prion disease. Um, if you look at the blue curves, these are animals that got ASO therapy. So this is a therapy that, you know, we worked with Ionis to develop. It targets the prion protein RNA, destroys it, and therefore there's less prion protein in the brain. The small blue dashes are animals that got treatment late in their disease course. But by late, I mean prions had already replicated and filled the brain, but it was only the very beginning of symptoms. Um, the long blue dashes are animals that got treated way before anything was going wrong in the brain. And you can see that in that early treatment paradigm, and I will keep returning to this because I think early treatment is like, those are the two most important words I ever say. But you can see that in that paradigm, we tripled the survival of the animals. So this, to me, is the kind of most powerful data that came out of all of that work. And on the back of this data and, many, many other experiments, 10 years later, basically, at the very end of 2023, ASO, lowering the amount of prion protein in the brain, reached first in human clinical trials. And there were 16 sites. They've now all enrolled. Maybe we will hear a top level result about how this trial went by summer of this year. So this is, there's the race to the first drug and the race to the best drug. This is our very first opportunity to gather clinical data on lowering prion protein in prion disease patients. And my fingers are crossed. I think we could learn a lot from this trial, no matter how it went. And at this point, we just have to wait and see. So I guess one thing I want to talk about is, why did that take 10 years? And between that first conversation with Ionis and the first dose that went into people, there were all of these steps, basically. You know, what exactly do we want the drug to do? The proof of concept studies we did in animals, candidate selection. So what's like the perfect drug molecule Ionis did. And then there's this tremendous amount of, you know, here I'm oriented around FDA because we're working within the US system, but no matter where you are, um, doing, all of the manufacturing and the safety studies and like the quality control that meets the standards of regulatory authorities. So target selection, I would say this is where we are very lucky in prion disease. So probably you have heard kind of discouraging numbers about how few drugs that reach phase 1 clinical trials ultimately get approved for patient use. Um, it's a really small number. It's less than 10%. But I don't let that discourage me too much, because I think a major reason for all of those failures is not having the right target. I think there are a lot of diseases where we don't actually know what molecule we need to go after to intercept the disease process. Um, if we happen to have a clear target, I think that gives us a big advantage. And I would say that in prion disease, we do. So, you know, I was able to receive a predictive genetic test report telling me that I'm at high risk for prion disease because this genetic blueprint was already worked out. We know that there's one gene, the prion protein gene. We know that it gives rise to one RNA, and there aren't a whole bunch of versions, there aren't a whole bunch of isoforms. It's really just one RNA that gives rise to one normal protein. That's normally not a problem, but that's capable of misfolding into this so-called prion conformation that has the ability to grab other PRP molecules and misfold those, leading to this sort of cascade across the brain, and is also toxic to neurons. So whether we're talking about genetic prion disease or sporadic prion disease that happens at random and will kill about 1 in 6,000 people, or we're talking about prion disease in other mammals, not humans, all cases follow this blueprint. And that has led our lab to focus our energy really, really closely on lowering the amount of normal prion protein in the brain. if you don't have this molecule, the rest of this pathway can't happen. There's a lot of evidence going back many years that lowering prion protein should be helpful and should be safe. So I won't go through all of this in detail, but basically, if you look at the brain of somebody like, an animal or a human who died of prion disease, you can see these abnormal prion protein deposits. We know from human genetic evidence that all of the families that have genetic prion disease, the mutation is in the prion protein gene. That's a really clear causal link. We know that if you knock prion protein out, like if you genetically engineer animals to not have any, you can't give them prion disease no matter how hard you try. You inject them in the brain with prions and nothing happens. So I guess what I'm always trying to tell our community and but other disease communities as well is the more lines of evidence that we have pointing us to a clear target, the more we can bias our odds of drug development for success. And anytime we hear a different, drug target being explored for our disease, I think we should ask for these categories of evidence. We need to be betting on the right things if we're going to get this done in our lifetimes. So this is, a very wordy slide that I won't go through in detail, but just to say, discovering the drug is only one piece of the puzzle. And something that we've come to appreciate over the years is just how many other puzzle pieces there are. For example, we've needed to develop assays, methods for measuring the amount of prion protein in spinal fluid, and that can help us monitor how much there is in the brain. We have done work looking at the mutations associated with prion disease in the prion protein gene and how much risk do they convey. We have a natural history study where genetic prion disease mutation carriers come to Massachusetts General Hospital and they donate blood and spinal fluid. And we can use those samples to ask, what does the disease process look like? And what is happening before onset? And, I will say, the answer appears to be that prion disease mutation carriers really are healthy, like completely healthy for a variable number of years or decades until almost right before the disease process starts. I think this is a fabulous opportunity to treat pre-symptomatically and keep people healthy. It's not the case that there is some, underground pathological process happening for decades. In fact, this seems to be a very rapid disease both before and after symptom onset. So what comes next? We have a first drug that has now been in a small human trial. We think, and we don't have the human data yet, but we think based on our animal studies that maybe it was lowering prion protein in the brain by 50%. How can we do better? And I think there are, a few different things that can mean. We are working hard to figure out what does the next drug look like? How can we lower PRP more aggressively, more durably? Distributing drug around the whole brain is one of our big challenges. Prion disease is a whole brain disease. We always want our drugs to be as safe as possible. There's also the question of how do we make the next drug not take 10 years? And I think that now that we have walked this path once, we can move faster. But also, I think a very important way we need to do better is to be testing drugs for ability to prevent and not just ability to treat. So you've heard already today that prion disease is exceptionally rapid once it begins. And I think if you're in my position, it is not very attractive, the idea of waiting for those symptoms to begin before you ever start a therapy. We're dealing with cells and circuits in the brain that really can't be rebuilt. And the way that we can honor, the complexity and the beauty of the human brain is to protect it in the first place. So I'll talk very briefly about two drug types that we are working on in our lab. These are not programs where we have a company partner. We are just trying to do this all ourselves. It's really hard. But On the left, uh, this is another kind of oligonucleotide drug, um, not very different from an ASO, but this one is a small interfering RNA. And basically, it functions very similarly. The goal is for it to bind the prion protein RNA and trigger that RNA for destruction so that less protein is ever made in the first place. Um, a nice thing about this particular format, the divalent siRNA drug type, it's very new, but it appears to have better distribution properties. It appears to last longer in the brain and lower prion protein more than any ASO that we've ever tested in our hands. So this is one up and coming therapy that, in fact, we hope might be able to reach the first patient sometime in 2025. On the right, this is like a high risk, high reward strategy, but basically we are also working on a gene therapy. Um, so what you're looking at, this little diagram, is a viral vector. And in order to carry a gene therapy to the whole brain, we basically need to put it in a package that can cross the blood-brain barrier. 5 years ago, there was no such thing. But we are super lucky to be doing this work in 2025. Last year, our collaborator, Ben Deverman, published a viral vector that binds the human transferrin receptor, which is expressed on the blood-brain barrier, and uses that receptor to enter the brain. So then the question is, what are we going to put inside this package? And our collaborator, Jonathan Weissman, has been working to develop tools that are basically able to go to the gene of interest, that particular segment of DNA, sit on the promoter that controls whether that gene is active, and shut it down. So the goal for something like this, although it is newer and in some ways, riskier, is that it could potentially be a one-time therapy. Once you inject an AAV, like a viral vector, the goal is that it goes to the target cells and it keeps making that therapy indefinitely inside those cells. So we are hoping that this could be a way to turn prion protein down indefinitely. So I'm just going to show one plot for each of these. And basically what you're looking at is 100% is the amount of prion protein that is normally in the brain. These are mouse data. And here you're looking at, on the left, mice that got a single dose of a divalent siRNA therapy. This is delivered into the spinal fluid. And you can see that 1 month after therapy, we were reducing prion protein in the brain by 70%. So that's more than we've ever seen with an ASO. I think that's really encouraging. 3 months later, it was still down by half. And even 6 months later, it had not returned to baseline. So any kind of oligonucleotide therapy, you're going to have to redose. And as of right now, you're going to have to dose into spinal fluid, which in humans means a lumbar puncture at the base of the spine. We don't want to be doing that kind of thing too often. I mean, it's safe, it's tolerable. I have gotten 7 lumbar punctures for research myself, and I can say it's actually not a huge deal. Nevertheless, the more we can minimize sort of the dosing, the better. And I think here we're looking at a therapy that you might only have to dose 2 or 3 times a year. On the right, these are the data from CHARM in the mouse brain. And you can see again, 100 is an untreated mouse. And these are 3 different doses where, again, at that highest dose, we're probably looking at 80% reduction of prion protein. And as I mentioned here, we're hoping this is actually a stable decrease. We have animal studies that are going out to 6 months and a year to see if this is maintained after a single dose. And so far, the answer seems to be yes. So on both fronts here, I am optimistic that although there is still a tremendous amount of work to do, these drugs are doing the right thing. And it's a question of how to get them to patients as fast as we can. These are both really unusual projects in the sense that we're a rare disease. We can't always count on having a company partner. For the ASO, we did, and that had pros and cons. For these projects, we don't. So we are basically developing novel drugs out of our own lab. That's not a very standard thing to do. Um, I would say a pro of this strategy is that how we try to push this forward is up to us. And, we're a patient scientist run lab, so we're going to try to push it as fast as we can. Um, we have been willing, I think, in a way that companies are not always comfortable. We've been willing to go to regulatory authorities and ask, how fast can we move this? How fast can we get this both to symptomatic patients and pre-symptomatic people at risk? So that part of the process, I feel really great about. Um, I would say the major drawback of developing drugs ourselves as an academic lab is just that it's really hard. It's just a tremendous amount of work, but we're lucky to have a great team. So this is bringing me kind of to the final point that I want to leave you with, which is that I think when I talk about being able to try to be aggressive on behalf of patients, I think one of the most important ways that we can do that is to make people think about prevention and to keep it front and center in the minds of everybody we work with and everybody who has the ability to help us. Um, I showed you earlier this plot on top, which has the long blue dashes for the animals that got treatment. This was the prion protein lowering ASO treatment before anything had gone wrong in the brain. So it's not just before symptoms, it's before you could see any kind of molecular damage. Um, and that is so powerful, treatment at that early stage. By the time you're like nearing the cusp of early symptoms, you can see in the small blue dashes that you can still do a benefit, but that benefit has been dramatically reduced in degree. And then if you look at the plots below, these are studies where we let animals go out into the symptomatic phase and treated after symptoms. And on the left, this is animals where if you looked in the cage, you could see that they were sick. They had all started losing weight already. They have impaired nest building. They're beginning to accumulate symptoms. If we treat at that stage, we can benefit some, but not all of the mice. And then if you look at this final plot, these are animals where some, shown in red, some animals in the cohort had already died before the treatment. So there's a distribution. Some have already reached endpoint, some have not. If we give a treatment, a single dose of treatment at this advanced stage of disease, we actually can't help any of the animals. So I return to this comparison often in my mind because I think there's a big translation gap in how we sort of discover drugs in the lab and then how we test them in people. And I think we have to be honest with ourselves that in a very, very fast moving, irreversible neurodegenerative disease, I'm not saying we're never going to be able to help symptomatic patients. I really, really hope we will. I think earlier diagnosis is going to be a big part of that, and it's something I'm advocating for. But to achieve this spectacular increase in survival, and not just survival, but healthy life, I think we need to intervene very early. And early means intervene in people who look completely healthy. This is a bold proposal. And when we think about clinical trials, we usually picture that we're going to treat people who are sick, um, and we're comfortable with that risk benefit. And then we're going to see who feels better or functions better or survives longer. But if we really want to do the most possible good, we need to be willing to treat people who are healthy in order to keep them healthy. And we know we can measure prion protein in spinal fluid to say, is the drug lowering the amount of the single causal protein in the brain? And if it is, we need to be willing to say, this drug is reasonably likely to predict clinical benefit. Let's treat people, let's follow them, let's gather the long-term data, but this is good enough to start doing the experiment of trying to keep people healthy. So at this point, I would say I'm optimistic in the medium term that we're going to have molecules that can lower the amount of prion protein in the brain, and that those molecules should be effective against our disease. This is what I'm fighting for every day. Where I feel our real jeopardy is in making sure that those molecules are given their best shot to help people. I'll say one more thing about prevention, which is in order to do it, we need to be able to find people before symptoms. And among people who are at risk for not only genetic prion disease, but other neurodegenerative diseases, who have a family history, who know for some reason that they might be at risk. It used to be the case 10, 15 years ago that a minority of people chose to get testing. Maybe in prion disease, it was maybe 25%. But we also know that those numbers are rising, in part because stigma is going down, which is really important, I think, in the era of, more widespread sort of acceptance of genetics. Um, and in part because I think people rightly perceive that there's more hope than there used to be, and that by getting involved, by getting tested, they can help make that, future where we have a drug a reality. A lot of people who have tested positive or are not yet tested but know they're at 50-50 risk are choosing these days to participate in our natural history study or in those who are run by other people around the world. And those data directly inform what kind of clinical trials we can do and directly inform our conversation with regulators about how soon we can do them. So I think, when I got my genetic test report, there were people who said to me, why would you get tested for this? There's nothing you can do. And I guess if there's one thing I, sort of close with, I would like for it to be that it is never the case that there's nothing we can do. We're fabulously lucky to be living in this era where the right kind of drug types are starting to exist on Earth. And if we really, really, sprint at full pace, I'm hopeful that we can marry those drug types to our disease in our lifetimes. With that, I'd just love to say thank you, and I'm happy to take any questions. Um, I can hear you fast. So if you don't mind repeating the question for me, that would be fabulous. Yeah, yeah. Sure. Yeah. Thank you. Fabulous questions. So the question of how early do we treat relative to prion challenge, we've done both. So we have done fully prophylactic treatment where we administer treatment before prion challenge, and then we've done prion challenge with treatment following by 1 week, 4 weeks, 8 weeks, 10 weeks, like the whole series. And basically what we see is that this kind of surprised me. I thought that absolutely prophylactic would be the most protective. But in fact, immediately after challenge is almost as good. And we don't really start to see the efficacy of treatment wear off until we start to see signs of neuropathology emerging in the mice. So this is about 75 days post-challenge that we can start to measure neurofilament light in the blood. And we know that neurons are beginning to accrue damage. They're releasing proteins. We can detect that. We can detect prion load that's rising in brain if we were to sacrifice the animals. At that point, the efficacy of treatment starts to go down. Um, so to me, this is a little was a little surprising to me to think that even after the first prions have formed, there's still an opportunity to do like really profound good as long as they haven't started to sort of enter the neurotoxic phase yet. Um, the second question, yeah, we worry a lot about making sure that we are not overlapping any like variable sites in the prion protein RNA. And I guess one thing to say is our therapy is not allele specific. So our philosophy is that 85% of prion disease patients do not have a mutation. They're just spontaneous cases at random in the population. And 15% of patients do have a mutation, but there's a variety of them. So instead of treating all of these people as ultra orphan diseases, we need one drug to lower all prion protein in a non-allele specific way, that like one drug to rule them all. Um, so what companies like Ionis have found is that actually their most potent ASOs don't tend to bind the protein coding region of the mRNA. They tend to bind in the UTRs. And so they're looking at, you know, these sort of non-coding regions where it's we can sort of map out the variability in the population and try to aim for stretches of sequence where we don't see much variation between people. So that's been the strategy so far. Oh, yes. Yeah, that's a great question. So yes, we've like done prion protein lowering studies across a bunch of species and models, including in humanized mice with human prion strains. Actually, earlier this week, Tuesday, we published a paper where we collaborated with David Liu's lab here at the Broad using a base editor to edit an early stop codon into PRNP. So it's mimicking the R37X early stop codon that we see in the population at some frequency. And that the survival study in that paper was done in humanized mice infected with sporadic CJD prions and E200K prions. And we see that by creating this early stop that reduces the amount of full-length prion protein in the brain on the order of roughly 50%, we extend survival in the humanized model as well. So we've done human prions. We've engineered actually knockout hamsters and knockout rats, and have done sort of both ASO studies and genetic studies in those animals, showing that if you have a heterozygous knockout, or if you have ASO treatment, you sort of extend life across these different models and strains. In mice, we've done, besides the humanized model, 5 different mouse-adapted prion strains, and we've also done a spontaneous model collaborating with Joaquin Castilla, using his TG vole mice. So basically what I see is that across all these models, if you reduce the amount of prion protein in the brain, you're going to do good. But I think the sort of quantitative answer can end up being different depending on the strain. And that's something that we're still unpacking a little bit. Obviously, in every case, if you go all the way down to 0, you're fully protected. But if you reduce to 50%, you get a greater fold increase in survival in an RML infected mouse than in an RRML infected rat. That could be that RML is just such a well-characterized model that we have sort of perfect disease endpoints. And for all of these other models that are less well-characterized, we're guessing a little bit, and we haven't defined the endpoints as sort of consistently and as clearly. Or it could be that genuinely there are some strain differences in how much you need to lower PRP to achieve a really spectacular benefit. To me, the mandate has always been that we need to push closer to 0. And I hope that's what some of these upcoming modalities are going to help us do. Yeah. Fabulous. Thank you so much.